Welcome, everyone. Today that's ISEE North American Chapter, are hosting our now annual SNRN lecture. This year, Diana PACYGA is our winner, so welcome. Very excited to hear from you. Diana holds a BS in molecular and cellular biology with a minor in chemistry from the University of Illinois. And she's currently a PhD candidate in human nutrition and environmental toxicology at Michigan State University. She combines human nutrition with toxicology and epidemiology in her research and has a long-term goal to investigate how prenatal exposures affect development in infants and children. So today she'll be talking with us about gestational phthalate, replacement exposure, a glimpse into maternal risk factors, biological targets, and gestational cardiometabolic health. So super excited for this presentation, and I will hand it over to you, Diana.
Perfect. Thank you for the lovely introduction. So I'm gonna turn off my video just in case my Wi-Fi gets unstable. Okay, so I'm assuming everyone can see my slides. And so, yeah, thank you again for the lovely introduction. And I'm honored that I was selected to present our research findings today at this North American Chapter and SNRN joint webinar. And so, like Joan said during the presentation, I will share some of my research that I've conducted over the past several years related to gestational phthalate and replacement exposure and women's cardiometabolic health. And my slides aren't, okay. So as an introduction to that, heart disease is one of the top 2 leading causes of death in midlife and older adult US women. And so this table here shows the top 6 leading causes of death in females, and the columns represent the different age groups. And so we can see that heart disease starts out as the 6th leading cause of death in females between the ages of 1 and 19. It rises to 4th between 20 and 40-year-old women, rises to 2nd in women between the ages of 45 and 84, and is the top leading cause of death in females over the age of 85. And so our group is interested in identifying the critical risk factors, particularly those unique to women associated with increased risk of later life cardiovascular disease. And so our group focuses on pregnancy, which is an important part of the life cycle, partly because it produces babies, but also, an aspect that is often overlooked until recently is that it happens to women. And pregnancy is an important determinant of maternal health, as more recent preliminary evidence indicates that pregnancy itself, or having adverse pregnancy or birth outcomes, including pregnancy hypertensive disorders, gestational diabetes, preterm birth, pregnancy loss, and inappropriate gestational weight gain, may have lasting consequences on maternal later life cardiometabolic disease risk. And so it's important to identify the risk factors associated with adverse maternal health, particularly during pregnancy. And so our group is interested in the impact of exposure to environmental chemicals on maternal health. So my aim today is to highlight 4 of our recent studies evaluating the impacts of exposure to one class of chemicals, phthalates, on maternal pregnancy health. So in part 1, I will discuss our findings related to determinants of phthalate exposure during pregnancy. In part 2, I will discuss our study evaluating associations of phthalates and gestational weight gain. In part 3, I will discuss our findings related to associations between phthalates and maternal metabolism. And in part 4, I will present findings from our study evaluating associations of phthalates with maternal estrogens. And so why focus on phthalates? Well, we know greater than 90% of pregnant women have measurable levels of phthalate metabolites in their urine. And so this suggests that pregnant women are widely exposed to these chemicals. And so this widespread exposure is because some phthalates are plasticizers, and they are used during food processing. They're found in food packaging. They can also be found in medical tubing and are in coatings of certain medications and supplements. And some phthalates are scent stabilizers, and so they're found in cosmetics and personal care products. And so because these chemicals are found in personal care products and cosmetics, women generally have higher phthalate metabolite concentrations than men. And phthalates are traditionally classified as endocrine disrupting chemicals based on experimental evidence showing that phthalates can interact with hormone receptors, particularly estrogen and androgen receptors, and therefore can alter cellular response. However, these chemicals are also considered to be metabolic disrupting chemicals. So as shown on this figure, experimental evidence indicates that phthalates particularly can interact with peroxisome proliferator activated receptor gamma, or PPAR gamma, which is an important regulator of burning and storing fat. They can also interact with liver X receptors, or LXR, which are important regulators of glucose, fatty acid, and cholesterol homeostasis. And they can also interact with retinoid X receptor, RXR, which have been shown to be involved in adipogenesis. And so it's because of these metabolic and endocrine disrupting properties of phthalates that we worry about exposure in pregnancy, especially since pregnancy is this sensitive window with dynamic changes in hormone and metabolic homeostasis. Additionally, studies in pregnant humans have shown that phthalate exposure in pregnancy is associated with adverse pregnancy and birth outcomes. Additionally, phthalate exposure in pregnancy has also been shown to be associated with child outcomes, including behavioral and cognitive problems, as well as childhood and adulthood obesity. But of interest to us was that there has been recent evidence showing that phthalate exposure during pregnancy is associated with maternal postpartum cardiometabolic changes, so including postpartum weight retention and changes in blood glucose and lipid levels after birth. And so this is why we conducted these 4 studies to further understand the impact of phthalates on maternal health during pregnancy. And so our studies use data collected as part of the Illinois Kids Development Study, or IKIDS, which is an ongoing prospective pregnancy cohort in Champaign-Urbana, Illinois. And the goal is to recruit 720 pregnant women. And so IKIDS is one of the NIH, EHS, EPA Children's Environmental Health Research Centers, and it's located in Central Illinois, just in case you don't know, and it's run through the University of Illinois, Urbana-Champaign. And so specifically, women and their children are followed from early pregnancy through childhood. And recruitment for this study began in 2013 and is still ongoing. But for the studies I will be discussing today, we included women enrolled through 2018. And this is the IKIDS prenatal timeline, where women are recruited at 8 to 15 weeks gestation. And then after the initial visit, we are in contact with women at 4 more time points, approximately monthly. And so at the baseline, or 8 to 15 week gestation visit, women provided information about their sociodemographic characteristics. So this includes race, ethnicity, and socioeconomic status. They also provided information about their lifestyle factors, including smoking and status and alcohol intake. They also filled out a food frequency questionnaire to ascertain diet quality. And women also self-reported reproductive characteristics, including parity and pregnancy intention. And so just to provide you with some information about what the IKIDS sample looks like, the sample is fairly homogeneous demographically, since most women were above the age of 30, 80% were non-Hispanic white, 86% were employed, 81% were college educated, and 71% had annual household incomes of $60,000. Also, most women did not smoke in the first trimester of pregnancy, and a little over half had a pre-pregnancy BMI below 25 kilograms per meter squared, so that classifies them as under or normal weight before pregnancy. And median diet quality, which was measured by the Alternative Healthy Eating Index, or AHEI, was 55.8 out of 110 points, but the range is pretty wide, suggesting heterogeneous diet qualities in this sample. And about half did not have a prior birth. And women also, during the prenatal window of the study, were asked to provide 5 first morning urines from 8 to 40 weeks gestation, as indicated on this timeline. And this was used to measure chemical concentrations. So just in case you are unfamiliar with phthalates, urine is the best medium for approximating exposure to phthalates, which are considered non-persistent chemicals because they are metabolized and excreted from the body within 24 to 48 hours of exposure. And so because of the short half-lives of phthalates in the body, urinary phthalate metabolite concentrations can vary drastically within an individual. And studies have observed weak to modest intraclass correlation coefficients between phthalates measured from urines collected within a day, across a week, and across months in an individual. And so given the high within-person variability in measured phthalate concentrations, we physically pooled the 5 urine samples for each individual, which is a validated approach for approximating total pregnancy exposure to phthalates. And the pooled samples were sent to the CDC for quantification of 15 phthalate metabolite concentrations, which are all listed here. And we use urinary metabolite concentrations to approximate exposure to phthalates. So for example, we are exposed to phthalate parent compound DHP, which is metabolized and excreted as 4 urinary metabolites. And so we can molar sum the concentrations of these 4 metabolites to approximate exposure to DHP. Other phthalate parent compounds, such as DEP, are metabolized and excreted as 1 urinary metabolite. And so again, we can use the concentration of that metabolite to approximate exposure to DEP. But a unique aspect of IKIDS is that we also measure DINCH and DHTP metabolite concentrations. And DINCH and DHTP are plasticizer alternatives that were introduced into the US market before or in early 2000s, but we have only recently begun monitoring concentrations of these metabolites in the general population. And these replacements may have similar adverse health consequences as the phthalates they are replacing, although the literature is very limited with regards to these replacements. And so in IKIDS, most pregnant individuals were exposed to all phthalates and replacements. So specifically, more than 97% of pregnant women in IKIDS had detectable levels of most phthalate metabolites. And metabolite concentrations in IKIDS were similar to those of same-age US women from the National Health and Nutrition Examination Survey, or NHANES. But I want to take some time to emphasize DINCH and DHTP metabolite concentrations in this sample, which is shown in this table. So specifically, greater than 77% of women had detectable levels of at least 1 DINCH metabolite, while all women had detectable levels of both DHTP metabolites. And if we compare DINCH and DHTP concentrations in IKIDS to those of a representative sample of same-age US women, DINCH metabolite concentrations in IKIDS were similar to those of US women, but IKIDS women had about 1.5 to 3 times the levels of DHTP metabolites than US women. And so this makes IKIDS a great population to study these replacements. So we've confirmed that there's widespread exposure to phthalates and their replacements in the IKIDS sample. And so this leads us to our first study where we wanted to identify the major predictors of high or low phthalate replacement metabolite concentrations in pregnancy. And so if you search on PubMed, there are many studies evaluating determinants of individual phthalate replacement and replacement concentrations in pregnancy. And these studies have evaluated sociodemographic characteristics, lifestyle factors, reproductive factors, and also measures of seasonality and time trends as predictors of these chemicals. And so we wanted to determine if these same characteristics are also important predictors of phthalate and replacement metabolite concentrations in IKIDS. And so just to specifically discuss the seasonality and time variables in IKIDS, we're using conception season as our marker of seasonality, and we're using enrollment year as our proxy to measure time trends. And so one issue with these previous papers is that pregnant women are exposed to numerous phthalates and their replacements. And so some more recent studies have focused on identifying determinants of phthalate mixtures using pattern identification methods, including k-means clustering and principal component analysis, or PCA. And so such approaches that identify predictors of mixtures may better identify pregnant women who will benefit most from interventions targeted at decreasing exposure to these chemicals. And so our objective was to utilize these pattern identification methods like k-means and PCA to evaluate predictors of phthalate and replacement mixtures. And so jumping right into it, first we use k-means clustering, which essentially categorizes women into groups based on their chemical levels. And we compared 1, 2, 3, and 4 group solutions to determine the number of groups that best represent chemical concentration profiles in IKIDs. And phthalate and replacement metabolite concentrations were specific gravity adjusted, and they were also natural log transformed and Z transformed. And the results I'm showing you will be based on a subsample of 309 IKIDs participants. And so overall, we observed that women clustered into 2 distinct groups based on their phthalate and replacement levels. And so in this graph, the y-axis represents the median concentrations in nanogram per mil, while the x-axis lists each chemical. And the points indicate the median concentrations of the whole sample, while the bars indicate the median concentrations of the specific group. So overall, women clustered, or women in cluster 1 or group 1 had lower median phthalate and DHTP concentrations than the whole sample. Whereas women in cluster 2 or group 2 had higher concentrations of all phthalates and DHTP than the whole sample. And so interestingly, DINCH did not meaningfully contribute to group membership. So excluding DINCH, we can call these groups as low all and high all. So next we use logistic regression models to evaluate the odds of having high all compared to low all concentrations. And these models were simultaneously adjusted for all the characteristics that are listed on this slide. And so specifically, marital status, household income, parity, conception season, and enrollment year were important predictors of group membership. So specifically, women had higher odds of high phthalate and DHTP concentrations if they were unmarried, had lower annual household incomes, and had more than 1 child. And women had lower odds of having high phthalate and DHTP concentrations if they conceived in the spring, summer, or fall, and if they enrolled later into the study. So next we used PCA, which is a method that identifies patterns among phthalate and replacement levels. So we are essentially grouping phthalates and replacements that track together. And we used eigenvalues and the total variance to confirm the optimal number of groups that best represent chemical concentrations in IKIDs. And again, phthalate and replacement levels were transformed as was done for the K-means analysis, and we are using the same subsample of IKIDs participants. So overall, we identified 4 groups of phthalates and replacements that track together. And this explained about 67% of the total variance in phthalate and replacement levels. And all groups were positively correlated with each chemical. So this means as group score increases, the chemical levels increase. So specifically, group 1 tracked with DINP, MCNP, and MCPP, which we called the plasticizer phthalate group. Group 2 tracked with MBZP, DBP, and DIBP, which we called the other phthalate group. Group 3 tracked with DINCH and DHTP, which we called the plasticizer replacement group. And group 4 tracked with metabolite MEP, which we called the personal care product group. So essentially, these chemicals are grouping based on common exposure sources. And then we use linear regression models simultaneously, again, adjusted for all the characteristics to evaluate determinants of these identified groups. And so just for simplicity, the following tables I will be showing only list the characteristics and categories that were identified as important. And each slide will present results from one of the identified groups. So starting with group 1, the following characteristics were important predictors of phthalate replacement levels, or plasticizer phthalate levels, sorry. So specifically, we found that women had lower plasticizer phthalate levels if they were Asian or of other race, if they were unemployed, if they enrolled later into the study. So this suggests that plasticizer phthalate concentrations are decreasing across time. And they also had lower plasticizer phthalate levels if they conceived in the spring, summer, or fall. And women had higher plasticizer phthalate levels if they smoked in the first trimester. So for group 2 or other phthalates, women had higher other phthalate levels if they were Asian or of other race ethnicity. If they had lower household incomes, and if they enrolled later into the study. So this suggests that other phthalate levels are increasing across time. And women had lower other phthalate levels if they had lower educational attainment and if they had lower diet quality. For group 3, or the plasticizer replacement group, women had higher plasticizer replacements if they were older, if they enrolled later into the study. So this suggests that plasticizer replacement concentrations are increasing across time, and if they had overweight or obesity before pregnancy. And women had lower plasticizer phthalate replacement levels if they were unemployed, if they had lower household incomes, and if they conceived in the summer. And lastly, for group 4, or the MEP group, women had higher MEP levels if they were older, if they were non-Hispanic, Black, or of other race, if they were unmarried, if they smoked in the first trimester, and if they did plan their pregnancy. And women had lower MEP levels if they enrolled later into the study, so suggesting that MEP concentrations are decreasing across time, and they had lower levels if they conceived in the spring or fall. So to conclude part 1, K-means clustering identified 2 distinct groups of pregnant individuals, those with high and those with low phthalate and DHTP levels. And PCA identified 4 groups of phthalates and replacements based on their common exposure sources. And so we identified several important sociodemographic, lifestyle, reproductive, seasonality, and time characteristics as important predictors of phthalates. And many of these associations are consistent with previous studies. But most importantly, our study contributes critical information about determinants of DINCH and DHTP. And so in case anyone's curious about some additional findings, these results were recently published in Environment International. So this study relates to our overarching objective, since many of the determinants of phthalates and replacements are also associated with gestational weight gain, maternal metabolism, and maternal estrogens. And so the characteristics we identified may be important confounders to consider when evaluating associations of phthalates and replacements, particularly phthalate replacement mixtures with maternal pregnancy health, including one important marker of pregnancy health, gestational weight gain, which is the second part of today's talk. And so again, gestational weight gain is an easy indicator of maternal and fetal health during pregnancy, and appropriate gestational weight gain is critical to support the health of pregnancy as well as fetal growth and development. And so deviations from appropriate gestational weight gain are associated with adverse pregnancy, birth, and long-term maternal health outcomes. So for example, women with insufficient weight gain have higher risk of delivering preterm, may be more likely to have a small for gestational age newborn, and may also be more likely to develop postpartum depression. Whereas women with excessive gestational weight gain have a higher risk of developing gestational hypertension, are more likely to have a large for gestational age newborn, and also may be more likely to retain their weight postpartum. But most importantly, gestational weight gain is a complex phenotype with contributions from both the mother and the fetus. So this figure here shows the composition of weight gain for a normal weight woman, or a woman who was in the normal weight category for pre-pregnancy BMI, who is recommended to gain between 25 and 35 pounds. And so overall, baby contributes about 6 to 8 pounds, while the placenta contributes about 1 to 2 pounds to total weight gain. And the remaining weight gain is related to supporting the health of the pregnancy by increasing fluid volume, increasing protein and fat storage, and expansion of tissues, so specifically breast and uterine tissue. And so given the importance of appropriate gestational weight gain, several studies evaluated associations of individual phthalates with gestational weight gain in pregnant women. But the current literature is somewhat mixed regarding the direction of associations, where associations differ by study population and phthalate. And so metabolites of DEP, DEHP, DBP, and BBZP were found to be associated with gestational weight gain. So just as an example, for major personal care product phthalate DEP, 3 studies found that DEP metabolite was associated with higher odds of excessive gestational weight gain. However, 2 studies do not support this association direction. And we observed similar conflicting evidence for DEHP, and additional studies are needed to corroborate associations of DBP and BBCP. But most importantly, limited studies assessed associations of DINCH and DHTP with gestational weight gain. And again, there's also this issue that pregnant women are exposed to multiple chemicals. And so on this figure here, if we individually look at exposure to chemicals from toys, cosmetics, and food and their association with adverse health outcomes, we may find that observed risks may not reach the level of concern, which is this dotted line. However, if we evaluate chemicals from all these sources together, which is represented by this gray line, the cumulative effect of the mixture could be greater than the individual effect, and this would cross the threshold of concern. And so there are statistical mixtures approaches that have been developed to better simulate real-life exposure. And so the benefit of these statistical mixtures approaches is that they can estimate the cumulative effect of multiple chemicals on an outcome of interest, but also identify bad actors or which chemicals contributed most to the mixture effect. And so our objective for this study was to evaluate associations of phthalates and replacements, both individually and as a mixture, with gestational weight gain. And also some studies suggest that gestational weight gain differs by fetal sex, so in women carrying males versus females. And this does make sense given that the fetus contributes to weight gain. So we also evaluated whether these associations differed by fetal sex. So to calculate gestational weight gain, women self-reported their pre-pregnancy weight. And ideally, we would also have the weight at the end of pregnancy so that we could calculate total gestational weight gain. However, in iKids, the study is still ongoing, so we do not have medical records, but we followed women closely and communicated with them frequently. So we obtained self-reported weight at their last obstetric visit before delivery, which occurred at median 38 weeks gestation. So instead, we calculated gestational weight gain through late pregnancy. And median gestational weight gain through late pregnancy was 15 kilograms. And because we were unable to calculate total gestational weight gain, we also standardized gestational weight gain by pre-pregnancy BMI and gestational age at late pregnancy weight using a representative population of pregnant women. So for our statistical analysis, we use linear regression models to evaluate associations of individual phthalates and replacements with gestational weight gain z-scores. And for our mixtures analysis, we use weighted quantile sum regression, or WQS, to evaluate associations of a mixture of 10 phthalates and replacements with gestational weight gain z-scores. And again, for all analyses, phthalates and replacements were specific gravity adjusted. And we accounted for important covariates that were included in previous studies evaluating these associations. And again, we assessed associations in all women and stratified by fetal sex. So for the results, I'll be presenting a series of tables where the first column lists the phthalate and replacement biomarkers, while the next column presents the change in gestational weight gain in 95% confidence intervals for every 10% increase in phthalate and replacement concentrations. So meaningful associations are bolded and shaded. And the top part of this table presents results from single pollutant models, while the bottom 2 results rows present findings from WQS. And just in case you're unfamiliar with WQS, a unique feature about this method is that you can only evaluate one association direction at a time. So in this table, WQS negative represents results from models evaluating associations in the negative direction, while WQS positive indicates results from models evaluating associations in the positive direction. And this mixtures method also allows us to see which chemicals contributed most to the overall mixture association, which I will be presenting in pie charts. So overall, we observed that only plasticizer phthalates, DHP and MCP, as well as replacements DINCH and DHTP, were meaningfully associated with lower gestational weight gain z-scores. And similarly, the phthalate and replacement mixture was also meaningfully associated with lower gestational weight gain z-scores. And this association is larger than those observed for individual phthalates. And we found that DHP, DHTP, DINCH, and MEP were the most prominent contributors to the negative mixture associations, where DHP contributed 23% to the mixture association, DHTP contributed 20%, DINCH contributed 18%, and MEP contributed 13%. So the next series of tables and pie charts are very similar to those presented earlier. And here we're just additionally presenting results for women carrying females. So overall, we observed that previous associations of DHP with lower gestational weight gain Z-scores were driven by women carrying females, as indicated by this stronger magnitude of association. And similarly, inverse associations of the mixture with gestational weight gain Z-scores were more prominent in women carrying females. And we found that DHP, MEP, and DINCH were the largest contributors to mixtures associations in women carrying females, where DHP contributed about 39% to the mixture association, MEP contributed 12%, and DINCH contributed 10%. And here are the results in women carrying males. So previously observed associations of replacements DINCH and DHTP with lower gestational weight gain Z-scores were driven by women carrying males. However, interestingly, in women carrying males, an additional positive association between DINP and gestational weight gain Z-scores emerged. And as a result, we observed a more prominent positive than negative association between the chemical mixture and gestational weight gain Z-scores in women carrying males. And we found that DINP, MBZP, and MEP were the largest contributors to the positive mixture association in women carrying males, where DINP contributed about 34%, MBZP contributed 21%, and MEP contributed 12%. So to conclude part 2, select phthalates and their replacements were associated with gestational weight gain through late pregnancy. And additionally, our mixtures associations of phthalates and replacements with gestational weight gain were somewhat consistent with single pollutant findings, but also suggest a unique patterns of associations. But most importantly, our mixtures findings support the additive impact of multiple phthalates and replacements on gestational weight gain. And also, interestingly, our fetal sex-specific findings indicate that associations may be more complex than indicated by previous studies. And so this definitely needs to be corroborated by future studies. And this manuscript for this study is currently under review and should hopefully be published soon. So given that we observed associations between phthalates and replacements with gestational weight gain, we next wanted to evaluate whether phthalates and replacements are similarly associated with maternal metabolism. And this is relevant since gestational weight gain and maternal metabolism are related, although the association direction is a little unclear. And so when I discuss maternal metabolism, I am referring to glucose and lipid homeostasis, which can be assessed by measuring metabolic factors such as insulin, glucose, cholesterol, and triglycerides, just as examples. And so one proposed association between gestational weight gain and maternal metabolism is that gestational weight gain alters maternal metabolism. And so this is based on studies suggesting that higher gestational weight gain, especially in early pregnancy, is associated with increased risk of developing gestational diabetes and gestational dyslipidemia. And altered markers of glucose and lipid homeostasis are underlying hallmarks of these conditions. But the reverse could also be true, where changes in glucose and lipid homeostasis may influence gestational weight gain. And so just as a reminder, gestational weight gain is this complex phenotype with contributions from not only the mother, but also baby. So studies have shown that gestational weight gain and dyslipidemia are associated with altered fetal growth, which is an important component of gestational weight gain, especially later in pregnancy. But regardless of the directionality of these relationships, metabolic adaptations are critical for supporting fetal and maternal health during pregnancy. And so our goal was to evaluate associations of phthalates and replacements with a large panel of maternal metabolic factors that are listed on the slide that are associated with gestational weight gain or have been shown by the prior literature to be associated with gestational weight gain. But instead of evaluating the metabolic factors individually, we were interested in identifying patterns among these metabolic factors using PCA to determine whether phthalates and replacements target specific biologic pathways. So in iKids, women provided a fasting blood sample between 13 and 22 weeks gestation. And serum aliquots were sent to the Michigan Diabetes Research Center Chemistry Laboratory for quantification of serum concentrations of the previously listed metabolic biomarkers. And then we used PCA to identify patterns or groups of maternal metabolic factors. And again, we used eigenvalues and total variance to confirm the optimal number of groups that best represent maternal metabolic concentrations. And again, metabolic factors were natural log and Z transformed. And the results all presenting are from a subsample of 300 iKids participants. So overall, we identified 3 groups that explain 73% of the total variance in maternal metabolic markers. And again, all the groups tracked positively with the maternal factors. So this means as group score increases, the metabolic marker concentration increases. So specifically, group 1 tracked with glucose, insulin, C-peptide, and leptin, which are all markers of glucose homeostasis. Group 2 tracked with cholesterol, specifically total HDL and LDL, which we call the cholesterol group, while group 3 tracked with VLDL cholesterol, triglycerides, and free fatty acids, which we called the lipids group. So essentially, the metabolic factors loaded based on known biological pathways. And then we used linear regression models to evaluate associations of the 10 phthalates with each group. And overall, we found that only DHP, DINP, DEP, and DHTP were associated with the metabolic factors. And so specifically, we found that DINP and DHTP were associated with higher levels of glucose homeostasis biomarkers. We also found that DHP was associated with higher levels of lipid biomarkers, while MEP was associated with lower levels of lipid biomarkers. And so to conclude part 3, select phthalates were associated with altered glucose or lipid homeostasis. But most importantly, we found that DHTP was also associated with altered glucose homeostasis. And so these findings confirm results from experimental studies that phthalates are metabolic disrupting chemicals, but also provide new evidence that DHTP may also have similar metabolic disrupting properties as the phthalates they're replacing. And so these data are preliminary and they were submitted as an abstract this abstract was accepted for presentation at ISEE 2022, so stay tuned. So just to tie this all in together, rather unexpectedly, our findings evaluating associations of phthalates and replacements with maternal metabolism went against what we hypothesized, given that we observed phthalates and replacements are associated with lower gestational weight gain in the overall sample. But when we did evaluate associations by fetal sex, we observed that phthalates were associated with higher gestational weight gain in women carrying males. And so we may need to determine if associations of phthalates with maternal metabolism also differ by fetal sex. But additional studies are needed to further understand what is happening between phthalates and replacements, gestational weight gain, and maternal metabolism. So finally, moving on to part 4, we evaluated whether phthalates and replacements are associated with maternal estrogens. And this is relevant since maternal estrogens are known regulators of gestational weight gain, and this is partly due to their important role in metabolic adaptations. So this diagram shows the increase in estrogens from 0 to 40 weeks gestation. And the gray lines represent the rise in estrogens, specifically estradiol, estrone, and estriol that occurs across pregnancy. And this rise in estrogens support numerous functions during pregnancy, but of interest to us is that they support metabolic adaptations. And that's how they regulate gestational weight gain. So specifically, estrogens regulate gestational weight gain by influencing appetite, where estrogens can interact with leptin and ghrelin signaling. Estrogens can also induce normal maternal insulin resistance, and this is important to shuttle glucose to the fetus, which is the preferred fuel substrate for the fetus. And lastly, estrogens can induce maternal fat accumulation to store fuel for lactation. And so our objective was to evaluate associations of phthalates and replacements with maternal estrogens. And given that a majority of maternal estrogens during pregnancy are placental derived, we also evaluated whether associations differed by fetal sex. And finally, given that estrogen concentrations differ across pregnancy, we also evaluated whether these associations differed by gestational time point. So because urine is easier to obtain than blood, especially at multiple points in pregnancy, our goal was to develop an approach to measure estrogens in urine, especially given that in IKIDS, women provided multiple urines across pregnancy. And so estrogen metabolism is shown in this figure where parent estrogens estrone and estradiol at the top are metabolized into estriol and 12 other estrogen metabolites. And so we used LCM-SMS to measure urinary estrone, estradiol, estriol, and 5 additional major urinary estrogen metabolites, which are bolded in this figure. And all urinary estrogen metabolites were specific gravity adjusted to account for urine dilution. And to take advantage of the repeated urine collections across pregnancy, for IKIDS, estrogens were measured at 3 time points, 8 to 15, 25 to 33, and 32 to 40 weeks gestation. And these correspond to gestational periods associated with low, medium, and high pregnancy blood estrogen levels. And at each time point, we summed the 8 measured urinary metabolites to approximate total pregnancy estrogens at 8 to 15, 25 to 33, and 32 to 40 weeks gestation. So how well do urinary estrogens correlate with blood? Unfortunately, women did not provide blood samples at the same time points we assessed hormones, so we are unable to fully validate this approach. But just as a reminder, blood estrogens show a pattern of increasing concentrations across pregnancy. And so on the right are IKIDS urinary estrogen concentrations, where the blue box is the 8 to 15 week time point, the red box is the 25 to 33 week time point, and the green box is the 32 to 40 week time point. And so we were excited to see that urinary estrogens also had the same pattern of increase across pregnancy as estrogens measured in blood. So this at least confirms that the approach of measuring estrogens in urine may be useful, especially when urine is more readily available than blood. So moving on to our main objectives, just as a reminder, our first objective was to evaluate overall associations of phthalates and replacements with maternal estrogens. And so we use linear mixed models to evaluate overall associations of phthalates with urinary estrogens. And so this table here, the first column lists the 10 phthalates and replacements, and the second column presents the percent change in maternal urinary estrogens for every 2-fold increase in chemical concentrations. So overall, we found that DHP, MCPP, DINCH, and DHTP were associated with higher urinary estrogens. Next, we evaluated whether these associations differed by fetal sex and by gestational time point. So we also used linear mixed models to evaluate these associations, and results were obtained from a 3-way interaction between chemical, fetal sex, and gestational time point. And so just for simplicity, the third column for fetal sex and the fourth column for gestational time point just state the sex and time point for which we observed the strongest associations. So overall, previously observed associations of DHP and DHTP with higher urinary estrogens were more prominent in women carrying females in mid to late pregnancy. Additionally, previously observed associations of MCPP with higher urinary estrogens were more prominent in women carrying males in late pregnancy, while associations between DINCH and higher urinary estrogens were observed in both sexes, but were strongest in mid-pregnancy. And we also found that MCMP, DBP, and DIBP associations with urinary estrogens also emerged. And specifically, associations were observed in women carrying females in mid or late pregnancy. So to conclude part 4, we observed that plasticizer phthalates and their replacements DINCH and DHCP were associated with urinary estrogens. And so this confirms that phthalates and their replacements are endocrine disrupting chemicals. And I did not show these results, but we did evaluate whether these associations are potentially nonlinear by categorizing phthalate and replacement concentrations into quartiles. But our quartile results generally matched our linear findings, where associations tended to be stronger at higher quartiles. But most importantly, associations were more prominent in mid to late gestation in women carrying females. And so these time point and fetal 6 specific findings corroborate previous experimental evidence that phthalates and replacements are likely targeting placental estrogen pathways. And so these findings were published last year in Environment International. So just to summarize, overall, in part 1, we identified several sociodemographic, lifestyle, and reproductive characteristics, as well as seasonal and time trends that were predictive of phthalate and replacement levels. And these same characteristics are also important determinants of maternal cardiometabolic health. In part 2, we found that a mixture of phthalates and replacements was associated with gestational weight gain through late pregnancy, and these associations were fetal sex specific. In part 3, we found that select phthalates and replacements were associated with changes in glucose and lipid homeostasis, which we know these metabolic adaptations are related with gestational weight gain. While in part 4, we found that select phthalates and replacements were associated with altered maternal urinary estrogen concentrations. And these associations differed by fetal sex and by pregnancy time point. And this is important since maternal estrogens are known regulators of gestational weight gain and are also regulating maternal metabolic adaptations during pregnancy. So what about future directions? So the most important next steps for these studies will be to understand, one, the relationships between gestational weight gain and maternal metabolism, especially given the time point we measured these maternal metabolic factors and what's happening in this sample, as well as relationships between maternal estrogens with maternal metabolism, especially since maternal estrogens were measured in urine. And also to see how maternal urinary estrogens, exactly what's happening between these and gestational weight gain. And we hope to further identify links between phthalates and replacements, gestational weight gain, maternal metabolism, and maternal estrogens, and understanding what's happening here. And additionally, our major goal is to understand the long-term implications of associations between phthalates and replacements and maternal metabolic health. And we hope to do this in our new study called the Illinois Metabolic Outcomes in Moms, or IMOM study. And our goal is to recruit 350 participants at least, and specifically iKids moms, 4 to 7 years after the iKids pregnancy to evaluate these long-term associations. And so with that, I just like to acknowledge a few people. So most importantly, my mentor, Rita Strakowski, who has been really supportive as I've been navigating and will be finishing up my doctoral program. And of course, my lab mates who've also been very supportive. Also our collaborators, who are all co-authors on the manuscripts and abstracts for the studies I presented today. It's been really great working with everyone who have a diverse set of expertise in learning a lot of these statistical mixtures methods from them. And I'd also like to thank my home department, the Food Science and Human Nutrition Department, and the Institute for Integrated Toxicology at Michigan State. Our funding sources are listed here, but most importantly, I'd like to shout out the iKids team for working tirelessly to collect all this data and organize it. And of course, our participants, because without them, none of this would have been possible. And thank you for coming and listening to me talk today. And I'd be happy to take any questions, or if anyone has suggestions or comments, that'd be great as well. So thank you.
Thank you, Diana. That was a really incredible presentation. So thank you so much for sharing with us. If anyone has questions, we're a small enough group. I think you can feel free just to unmute and go ahead and ask. Otherwise, go ahead and type in the chat and I can read out any questions that we might get.
Thank you, everyone in the comments.
Yeah, really nice comments. It was very comprehensive, so perhaps folks are having trouble coming up with questions. I'll give people a few moments here, though.
I'll ask a question. Great talk, Diana. This is Sheree Farzan from USC. I'm really interested in your findings. I'm curious if you've had a chance to look at any other sort of pregnancy complications. Like, have you looked at any of the outcomes that might be related to some of these, I guess, intermediates, like these biomarkers of gestational weight gain, such as gestational diabetes or gestational hypertension or anything along those lines?
Yeah, so that's a great question, and we're definitely thinking about that. We currently do not have access to medical records, so we're unable to determine how many women had gestational hypertension, but also just to confirm the number of women with gestational diabetes. But this is a pretty healthy population in that aspect. So yeah, I don't know if we'll have enough people to be able to actually evaluate those things. If anything, it'd be great to maybe exclude these in sensitivity analysis just to see if some of our results stick. But yeah, we'd probably not be able to do this in this population, unfortunately.
Yeah, no, I understand. And now that you say that, I remember you said that earlier, that you didn't have access to medical records, but really interesting. Thank you.
So just an update on medical records. It's in process now. There's a big medical record abstraction project going on over the summer and into the fall. And we do have medical records for quite a few of the women now. We just don't have all the data abstracted yet. So we are pretty optimistic that by January, we should have most of the data from medical records available. So I agree with Diana. It's a pretty healthy population, but we will be able to look at some of those things fairly soon. So finally making progress on that front.
Great. Well, I'll stay tuned then.
Thank you.
Great. We have a question in the chat from Krista Christensen. Sorry if I missed this in the presentation, but can you briefly describe the validation study you mentioned with respect to pulling urine samples across pregnancy, please?
Yeah, so there have been a few studies out there collecting multiple urine samples from pregnant women across days, across months, and across weeks, just to determine how variable concentrations are even within one individual. And so there was one study that also then pooled about 4 or 5 urine samples as well. And then they evaluated associations of chemicals measured in these urine samples, either spot or this pooled, with certain outcomes. And they ended up finding that there were a lot stronger associations and potentially missed associations if urine wasn't pooled. And so that suggested to us that at least this would be important, at least for non-persistent chemicals, because we may be missing a lot of that exposure if we only relied on that one urine sample, because that one time point might not be representative of if a woman showered the previous day and what products she used or what food she ate, and lifestyles are very different across even days and weeks. And so, yeah, and so that study was really important. And that's why we decided to also pool. But thank you for the question.
Any last minute questions from anyone? All right. Well, Diana, again, thank you so much. This was very impressive, and we will be uploading the recording to ISEE's education YouTube channel for folks who weren't able to be here today. So, and thanks to everyone else for attending.
Thank you, everyone.